Pauling and Corey predicted a second type of secondary structure which they called beta sheets, or generally they call that beta conformations. This is a more extended conformation of polypeptide chain as compared to alpha helix. So in this beta sheets, the backbone of the polypeptide chain is extended into a zigzag structure. So a polypeptide chain that is arranged in a zigzag manner, and the zigzag polypeptide chains are then arranged side by side. For example, a one polypeptide chain that is zigzag, and the other one is also zigzag, and these two chains, they are arranged side by side. So when they are arranged side by side, they resemble a pleated sheet. That's why these structures were called as beta pleated sheets. So here you can see these three polypeptide chains, 1, 2, 3, and these three chains are arranged in zigzag manner. And when these three chains, zigzag chains, they arrange each other side by side, they make a sheet. And in this sheet, these, you can see, these are the pleats. So this is called beta pleated sheet, in which the chains are arranged side by side. In this sheet, the R groups in the adjacent amino acids, they protrude out from the sheet, from the zigzag structure in the opposite directions. So if this is a polypeptide chain, the R groups lie here, here, and here. As you saw in case of alpha helix, the R groups, they lie outside the helix. So here too, the R groups lie outside the sheet. This is because the R groups may not create any sort of hindrance. As you can see in this demonstration, this is polypeptide chain which is zigzag arranged, and the R groups, they lie outside. They are actually protruding out of this sheet. And when another polypeptide chain will come to the side of this, and like this, they will also have their R groups protruding out. So these R groups will not create any hindrance in joining these polypeptide chains. Hydrogen bonds are formed between adjacent segments of polypeptide chains. So when two zigzag polypeptide chains come close to each other, hydrogen bonds are formed between different segments of these two polypeptide chains. The adjacent polypeptide chains in a sheet can be either parallel or anti-parallel. So this arrangement of polypeptides is either parallel or anti-parallel. What is meant by parallel and anti-parallel? First, let us see the arrangement of hydrogen bonds. So here you can see this is one polypeptide, this is another. They are arranged in a zigzag manner, and the hydrogen bonds here, you can see these are the hydrogen bonds, and these are present between the adjacent polypeptide chains. So this is anti-parallel polypeptide chain. Anti-parallel means at this corner, you can see this is amino group, and on the other chain here is a carbonyl group. If you see on the other side, same, one is amino group and the other one is carbonyl group. So the direction of these polypeptide chains is like this. One is moving in this direction and the other one is moving in this direction. So this is called anti-parallel beta pleated sheet. If we see the parallel beta pleated sheet, you can see amino group. Both are on the same side. So the direction of these polypeptide chains is parallel. Look at the difference between the hydrogen bonding in antiparallel and parallel beta pleated sheets. The hydrogen bonds are comparatively straight in case of antiparallel beta pleated sheets, while they are slightly distorted in case of parallel beta pleated sheets. That's why anti-parallel beta sheets are more stable as compared to parallel beta pleated sheets.